Good, good. Anybody?
Okay. Next is Paul Marquez. Who's going to be talking. Marquez?
Yeah.
Oh, so he's going to be talking about the same dose of nicotine induces condition, place preference, or aversion, the importance of tactile cue.
Now we're going back into the laboratory. The clinical back into the lab. So some of you may already know some of these techniques. How do I, okay. Sorry, I have to do this. Okay. Got it. Okay. So basically what we see in the lab is this is a CPP paradigm, pretty common. I think some of you know of it. Some may not be aware of this, but it's in looking at animal studies. And what we've done is we were specifically focusing on only CPP, and that's condition place preference. Now, we were also looking at, of course, a similar dose of CPP. Okay. So as we know, of course, nicotine addiction is a major public health problem. What we notice is that nicotine has, of course, a rewarding and also an aversive effect in the laboratory when we are looking at animal behavior. Get rid of that little thing there.
Right there, but okay.
What happened?
There's a little thing blocking your top, but
that's all right.
Okay, don't worry.
That's okay. I'll get to work around it. So with that being a rewarding and aversive effect, we've seen that previous studies have shown nicotine to show a bell-shaped response, meaning that at low doses to higher doses you get a peak response, and then you get a tailing off of more of a less response with higher doses. So what happens in the laboratory? It's widely accepted right now that in the lab, low doses of nicotine produce condition place preference, or an animal show preference for. I'll be going over specifically that condition place preference. But on the other hand, higher doses of nicotine have been shown to be aversive, and CPP versus CPA, and we'll talk about that next here. So what is this CPP paradigm for those that do not know? Basically, what you have is you have this arena, which is comprised of a tactile cue, which is an environment and which is different than the additional part here. In this case, there's a rod shape or bar shaped tactile, and this other one has a grid-like pattern. This is considered a neutral pair chamber. So normally what we would do is put an animal here, a mouse or something, one at a time, and we will allow it to free roam between the two chambers and grab a baseline data for the animal. And this is the textbook definition for that, so I'm describing it in another fashion. It does reflect as reinforcing the degree to which the animals can spend time in an environment. And like I mentioned, so you're comparing two environments. You have this environment, which is grid-shaped, and also you have this environment, which is a bar shape. And this is just tactile, one of many cues. There's visual cues you can tell by the different sort of colors, and then also you can incorporate other additional cues. In this case, we're looking at tactile cues. The procedure is really straightforward and pretty technically simple and usually brief, so you get a lot of information in a short period of time. Normally, what we would do, and I'm going to go a little bit more into the procedure, is that you would place an animal, again, here, and then we would, once we establish the time step between all three chambers within this arena, what we would do is we would isolate each arena, or each chamber, and then we would either condition the animal, which is hence the place preference. We would condition the animal in one side with either a saline or treated with, of course, nicotine, and then the opposing side we would choose another subject to do the same thing. So we're going to look at the drive between the two different environments. Yeah. And the condition can be at any schedule. You can do a short-term, long-term, acute, chronic. So what we did in terms of the CPP, this is the old one. It's okay. Okay, so again, what we do and what we did in the laboratory is we took some animals and we did a 1-week type of conditioning CPP. We looked at first the baseline, we let the animal roam for 15 minutes. Then we, of course, grabbed the sort of time which is spent through all 3 chambers. Once we did that, then we established which chamber we're going to condition them in, of course. So on the second day through the 4th day, we would either, again, once I mentioned, we would treat with nicotine and or saline in each chamber. So just like on day 1, where they're allowed to free roam, after the 4 days or 3 days of conditioning treatments, so for example, in this case, they would receive like saline for one animal. Let's say they were assigned for drug pair chamber in the bar. They would receive nicotine, a low dose nicotine here, or we would give them saline here. It could be on the given day, or we can go every other day or a longer schedule. In this case, it was twice a day, once in the morning and once in the afternoon. And then after 1 week's conditioning, on Friday, or the final day, what we would do is just read what happens on day 1 to see if there's 2 events. One, we're either going to see an aversion to one side, or we're going to see CPP to another side. And this is what we need to see, which is this one here. Okay, so our first thing to look, we're trying to determine whether or not a low dose of nicotine at 0.25, which other labs have found to be rewarding, to see if it induces CP or CPA. So what we're looking at is our paradigm. And if this response is dependent on that tactile cue that it's paired with for each bar or grid side. So on day 5, what we saw, the test day, is we noticed that if the animal was conditioned in this grid-like pattern chamber side, it showed a nice time response for time spent in that drug pair chamber. So there was a CPP event, which there was a preference for that side versus the saline and or vehicle pair side. So this was a preferred tactile cue. The second one was, of course, completely the opposite. What we've seen is that, and this different tactile cue, which is just bar shapes, they showed an aversion, even though in presence of a low dose, the same dose of nicotine. So with the context, there's a difference in the context with the same dose. So with that in mind, what we wanted to do is we wanted to see if we could take a look at that further. So we said, well, okay, we did this for 1 week, and now we're going to go on a little bit longer for possibly 2 weeks chronic. Smoking, obviously, nicotine use is a long-term event, and probably we noticed that it's kind of a weaker reinforcer. If we were to use morphine, it would be after a single dose, you'd see the animal spend a lot of time in either chamber. Okay, so we wanted to look to see if there was going to be a tolerance that developed to nicotine, induced CPP. So we chose, of course, the CPP side, which was, of course, the grid pattern. So we wanted to see if there was going to be a tolerance that developed, and if administration of alcohol, which we're getting back to the drinking and smoking ideology, of alcohol along with nicotine, would induce a greater CPP response, or does it show just the tolerance? And let's see what the mice will do. Is correct. Okay, so again, like I mentioned, the schedule was the same, but what we did is we did a second week of conditioning, and we did, of course, the nicotine alone, and then in conjunction with ethanol. And that's basically what it says here. Mice received 3 more twice daily conditioning, which was the second week. So here they are, the 2 weeks. If you look at just nicotine alone, day 1, of course, is the baseline day. But you don't see a preference for tactile on the baseline day, which is interesting. know that there's not an initial adverse effect to the tactile. But what we see, of course, in the grid parachain, we see that same similar response to the in the tactile, the preferred CPP that we've seen in the previous week. So it maintained for the first week, and then we went to the second week, and we noticed that slightly we went to similar baselines. So we're seeing possibly a tolerance that builds up to nicotine just in 2 weeks alone. But when we combine the two, alcohol, a dose of alcohol 1E1, and again the same dose as well, we see that similar baseline, and then we see a slight potentiation here, and or this similar, very slight potentiation in drinking. So you do see that smoking and drinking component becomes rewarding in that rewarding tactile area. Secondly, we will second we can see a stronger potentiation, but we can tell that also that the tolerance is affected by the alcohol. So again, it appears that mice show a similar preference for the mesh and rod prior to conditioning, like I mentioned. It's at a similar baseline for the two chambers. Again, however, depending on the tactile cue, mice either prefer the chamber associated with nicotine administration or avoided it. Again, we noticed that the tolerance had occurred in a 2-week regimen of nicotine, but alcohol prevented the tolerance phenomenon. Am I running out of time? Okay. Okay, so what we're going to look at next with all this in mind, so where it's going to go is studies have started. We're going to look at just the paradigm in general. Is there that initial preference for just saline, and maybe it's just the conditioning cycle for 2 weeks of only saline, looking at the mice to see if there's something about the paradigm itself. It's really kind of challenging the paradigm because a lot of research is based on these paradigms for behavioral studies in animals of addictive reinforcing drugs. We also have started, of course, alternate cues, and this maybe will hand in hand a little bit with e-cigarettes, because I know that e-cigarettes have introduced, they have flavonoids in them, like fruit flavors and also nutty flavors and so forth, association with them. So we're looking at possibly looking at olfactory cues that are associated with it. So if you are taking nicotine and you have a fruity flavor, is that also going to show a higher strong potentiator response? So we're currently working on that as well. Again, we were going to look at it a little bit longer for more conditioning cycles to look at a long-term context for that long-term smoker and or drinker, which we have seen in the previous presentation. And I think that's all I have. Right. Okay. And excuse me, don't throw tomatoes. I'm kind of new at this stuff. Any quick question? Any quick question?
Anna? Nope, it's up to you.